So you're walking down the aisle in your local health food store, and you're walking past the protein section, and you're seeing all these different options: whey protein, plant protein, all sorts of different things. And you instinctively go and buy a container of whey protein. And then, you leave the store and you're still doing you still have groceries to get. And so you go to the grocery store and you're getting ready to make dinner for the evening. So you're like, okay, I need chicken or salmon or something. And when you're buying your chicken, you ask yourself, is it really true that animal protein is easier to digest than plant protein? And what actually is the biomechanics of protein metabolism? And along with that, you also ask yourself, and I heard some people saying or talking about a 30-gram protein limit. What is that? And is that actually true? Well, let's talk about the science behind that.
Welcome to The Science Behind That with Atticus Hamilton.
Hello, all you scientists. Welcome on back to The Science Behind That podcast. I'm your host, Atticus Hamilton, and as you heard from the intro, today we're going to be talking about protein metabolism, protein biochemistry, and at the very end of the episode, if the 30-gram limit is a real thing or if it's just a bunch of hocus pocus. So, we're definitely going to jump right into it, but before we do, ladies and gentlemen, go grab yourselves a nice hot cup of coffee, add a little bit of maple syrup to it, and we'll jump right in. So, ladies and gentlemen, this is going to be an in-depth episode. I'm recording this now, and as of right now, I'm going to say it's probably going to be about an hour to get through, everything we have to talk about. So before we do that, I want to lay out really quick almost a table of contents, basically, of what we're going to talk about. So the first thing we're going to talk about is just basic protein biochemistry. Now, I know some of you out there are probably like, Atticus, I already know this. I took biochem in college or whatever. Don't do this. If that's the case, you can skip ahead. But this will be the first little section just where, I go over basically an overview of the biochemistry of proteins, how they're made, what they're made from. And that is actually really important later on in the episode when we talk about protein bioavailability of animal protein versus vegetable protein, and protein stereochemistry, because I've alluded to that a lot in a lot of my episodes, and I think it's finally time for me to directly address it. After that, then we'll talk exactly about that. After we talk about the basics of protein biochemistry, then I'm going to take everything that we had just talked about and sort of apply it to metabolism. And hopefully by the end of that section, you guys will be able to comfortably say, yes, I understand what protein bioavailability is, A, why it's not a myth, B, and the things that I can do to maximize it. And yes, there are people out there who believe it's a myth for some reason. And then after that, I'm going to wrap this episode up with a talk about the 30-gram protein limit. So this topic idea for today was sent to me by one of you lovely listeners out there. I got an email last week saying, hey, Atticus, I love your show, but I had an idea, and my idea was, could you talk about protein metabolism? You allude to protein metabolism and bioavailability and the biochemistry of these molecules a lot in a lot of your episodes. Could you do an episode specifically where you talk about this and maybe also talk about the 30-gram limit? So stay tuned for that as well. So thank you to that listener who sent me the email. I hope this answers all of your questions. And thank you for everyone for tuning in. So, starting off, the basics of protein biology and biochemistry. So, how are proteins made? Well, proteins, just like most things, come from a cell's genetic code. Now, before I get emails from you saying, Atticus, that's not always the case, I understand. So there are certain situations with certain organisms like bacteria, archaea, or yeasts that receive or ingest plasmids, and sometimes they may or may not incorporate those plasmids into their own genome. A lot of times, plasmids will come with their own polymerase, their own DNA and RNA polymerase, and they will just auto-transcribe and translate proteins, basically, within the cell of that yeast or bacteria or archaea. We're not going to be talking about those because the molecular genetics and the biochemistry of bacteria and certain eukaryotes like yeast can get very, very complicated very quickly. So for the purposes of this episode, we're just going to take a human cell. So human cells are monocystronic. Now what that means is monocystronic means that human cells encode one gene per mRNA, which is different than bacteria or prokaryotes, which are polycystronic, meaning they encode multiple genes per mRNA. And again, we're going to omit them from this discussion because they make things more complicated. So, looking at a regular human cell, you have DNA in the nucleus, right? And let's say the cell needs to transcribe and eventually translate a gene, right? So the section of DNA where that gene is located is going to be unwound and separated, right? Because DNA is double-stranded. And then one of those strands is going to serve as the template for the transcription into mRNA. So think of the DNA as the master blueprint and the RNA as, a photocopy of that master blueprint. So once that mRNA is produced, that mRNA is going to be shuttled out of the nucleus into the cytoplasm. Now on this mRNA, we have a couple things going on. Of course, we have the gene, right? But there's also a start codon, a promoter region, and a stop codon. Now, what is a codon? A codon is just 3 base pairs, and base pairs being adenine, thymine, cytosine, guanine, and uracil. Now, there is no thymine in RNA, it's uracil, but anyway. So that's a codon, and you can go and look up codon tables. They're anywhere on the internet. We're not going to get too deep into the specifics of codons and what every codon encodes for because each codon encodes for a different amino acid. Now there is some overlap, and so you could have multiple codons coding for the same amino acid, and that gets into silent mutations basically, and other mutations of codons, but again, that's more unrelated for what we're going to be talking about. So basically what happens is you have this mRNA, right, and it's in the cytoplasm, and also in the cytoplasm there are tRNA molecules, and tRNA stands for transfer RNA, and these tRNA molecules carry an associated peptide or amino acid. The amino acid is bound chemically to the tRNA because it's associated with the codon that tRNA encodes. And so basically what will happen is the ribosome will attach to the mRNA molecule, and it will read the sequence of bases in that gene on that ribosome. And it will bring in, essentially, I'm really trying to simplify this. It'll bring in the tRNA with the amino acid it needs, It'll bind that tRNA to its associated region on that mRNA, and then the amino acid that is on there will shift to the end, and then a new tRNA can bind, and then the amino acid that's on that will shift on top of the first one, and so on and so forth, until you have a very long strand of amino acids, right, hanging off of the last spot on this ribosome, because there's 3 binding sites in a ribosome. It's hanging off the last binding site in this ribosome, right? And once the ribosome hits the stop codon on the mRNA, it stops, and the amino acids are cleaved off of the ribosome using a peptidase, and then you have a peptide chain, and that is the primary protein structure, a polypeptide chain. So with protein structures, there's 4. There's primary, secondary, tertiary, and quaternary. Primary is that polypeptide chain we talked about. Now, it doesn't end here, and I can't really think of any proteins off the top of my head that do end here, that are just a polypeptide chain. There may be 1 or 2, but I can't think of what they are. Most proteins are going to be in a tertiary form. But the next stage is the secondary protein structure. Now, what does that mean? Well, the secondary protein structure is basically where the polypeptides, or the individual amino acids in that polypeptide chain, are hydrogen bonding with one another. And they hydrogen bond in a very unique way according to their stereochemistry. And this is why we had to talk about this first, because this is where the stereochemistry comes into play. But depending on the amino acid profile of this protein, that's going to change how they hydrogen bond with one another. But they can hydrogen bond into a secondary protein structure into 2 possible configurations: a pleated sheet, or a beta pleated sheet, or an alpha helix. And that's hydrogen bonding of the peptide backbone. And so it causes these amino acids to fold into a repeating pattern that can either be a pleated sheet, which just looks like a zigzag, or an alpha helix, which looks like a helix of DNA. Now, there are some proteins out there that stop at this point, right? So there are some proteins out there that in which their active form is a secondary structure. Not a lot, but there are some. Some of the ones I can think of are responsible in immunology, right? There's a whole host of immunoglobulins that are responsible for immune cell signaling that are secondary structures because they're smaller and they're easily transferable out of the cell. So one thing that we'll notice is that the more complex a protein becomes, the bigger it is, and the harder it is to move places, right? So a protein in a secondary structure form is going to be a lot easier to get out of a cell than a protein in a tertiary or quaternary form. But next, moving on to tertiary. Tertiary is where the protein continues folding, again, due to these hydrogen bonds, but now these chain-to-chain interactions of the beta-pleated sheets and the alpha helices to form a 3-dimensional structure that, I can't really describe the look of it. It changes based on the protein, but it develops a unique structure because of the hydrogen bonding now of the secondary structure. And most proteins are going to be active in this tertiary form. Now, what is a quaternary protein structure? A quaternary protein structure is essentially when you just have multiple tertiary proteins that have bound to one another. And that is the most complex form, right? So, if you have 2 separate proteins that are in a tertiary form, if they bind together and once they bind, they become their active component, then they are a quaternary protein, or that is a quaternary protein. And of course, this folding is determined by the composition of the protein itself and what amino acids are where. and that's going to determine how it folds. And then, of course, the specific amino acids that are in a protein is determined by the genetic makeup of the protein itself. What gene did it come from, right? What is the genotypic expression of this? Anyway, that's protein folding and whatnot in a nutshell. So that's part one. I hope that made a little sense. I hope it wasn't too confusing. But that's going to be very important for our second part here. But before we jump into that, I want to talk about stereochemistry. Right? Now, I've talked about this a lot. Now, what is stereochemistry? Well, essentially, all stereochemistry is it's the specific orientation of atoms within a molecule, right? So, you could have so take a carbon, right? Generally, it applies to a molecule where you have a center that is a carbon atom, right? So an organic molecule. This applies primarily to organic compounds. Now, I think a lot of people think, okay, well, take water, for example, or no, glucose. Take glucose, for example. Glucose, the formula for glucose is C6H12O6, right? 6 carbons, 12 hydrogens, 6 oxygens, right? C6H12O6. Most people, I think of a compound like that and just think, okay, here's the formula, so the configuration of atoms doesn't change. It's just one thing because that's how it's written. But that's not the case. And in fact, in organic chemistry, this happens a lot. And this is stereochemistry, where we can have two molecules that will have slightly different forms in terms of orientation of, let's say, a nitrogen group and a hydroxy group attached to a carbon. They'll have two different forms, but they'll be the exact same molecule. And this is called the D&L system. So the D&L system is when you have a molecule that looks like basically a mirror image, like you could fold it onto one another. That's called D&L. And I don't want to get too deep into this because it can get confusing very fast. But ultimately what this means is one amino acid from one source can be a D, for example. Let's take L-glyceraldehyde, all right? Or I shouldn't have said L-glyceraldehyde. See, now you know what its conformation is. Let's take glyceraldehyde. You could have a glyceraldehyde from milk, right? And the conformation of that is going to be an L, right? So what that means is, so glyceraldehyde, you have a hydrogen attached to a carbon, and then you have another group called a C2OH group, and that's attached to the same carbon. And in the L group, or in the L conformation, those two project to the right, and in the D conformation, they project to the left. So they're what's called a mirror image, or they're what's called an enantiomer. I always thought of it as a mirror image, but it's more of like, if you fold it on top of one another. Anyway, so L and D, it's the same amino acid, it's glyceraldehyde, but the one from the milk is an L-glyceraldehyde, and the one from the plant is a D-glyceraldehyde, assuming plants produce glyceraldehyde. But so you may be thinking, well, what does that matter, Atticus? It's the same compound. It's going to bind the same. It doesn't. And this is where bioavailability comes into play. For the most part, animal sources are going to all have a very similar stereochemistry with their amino acids. And it's going to be a little different based on amino acid. So for animals, for the most part, it's going to be an L conformation for glyceraldehyde, and plants is going to be more of a D conformation. But what this means is this actually affects protein folding, because you remember what I said, how the way we get from a primary protein to a tertiary protein is essentially hydrogen bond folding. Well, if you have a protein, it could be the exact same protein, but the amino acids in it are an L conformation from one source and a D conformation in another, the folding is actually going to be different. So the tertiary structure is going to look different in the one from the D source than it is in the L source. And this is how we end up with milk having a bioavailability of 100% and green pea protein having a bioavailability of 50%, because they contain amino acids that are enantiomers of one another. And there are certain enantiomers that the human body, unfortunately, cannot utilize when it comes to amino acids. And so what happens? They're excreted. Now, there is another aspect to this, which is optical rotation, where you can have basically, if you have an even split of an L and a D, or an even split of enantiomers, you can have what's called a racemic mixture. And we'll see that in a bit when we talk about certain protein sources. Certain protein sources give us around a 50% bioavailability, making the amino acids from them more of a racemic mixture of amino acids. But that's a little more out of the scope of what we're talking about here. What I really wanted to emphasize with the stereochemistry here is this is why bioavailability matters, and it's why it's important. So if you need to take a break for a minute, I'm going to pause right here while I get a cup of coffee before we transition into part 2. So I'll be right back. All right, hello everybody. Welcome on back. I hope you guys are ready to jump into part 2 here. So we talked in part 1 about the basics of protein biochemistry, and we touched on protein and amino acid stereochemistry. And now we are going to put all that together into the actual metabolism and how it affects bioavailability of these proteins. So what you guys really came here for, I guess. I don't know. I just enjoy that. I'm glad that you guys did come here, and I can maybe teach you guys something today. But so what is bioavailability? Well, bioavailability is basically just a measure of how much of a substance is able to be utilized by an organism, in this case, people. A lot of times when we talk about bioavailability, a lot of times we are referring to pharmacokinetics in pharmaceuticals, right? And the big reason for that is very important. So different pharmaceuticals have a different rating on, the bioavailability of their product, along with the duration that product is going to remain in suspension in the blood or wherever it needs to be. And proteins is no different. Now, on the internet, you'll find a lot of people who will try and tell you that protein bioavailability is a myth. I actually saw it was a blog, but still, I saw a blog where somebody was saying it was a myth, and they were a doctor. They were a registered dietitian. And to me, that was really shocking because it's not accurate, unfortunately. And it's inaccurate for two reasons. The first reason being, even with plant protein sources that are at 100% bioavailable, they're not the same as their corresponding animal protein sources, because animal proteins are complete proteins and plant proteins are incomplete proteins. Now, what do I mean by that? What I mean by that is there are 20 amino acids that are important for metabolism. In adults, 12 of those are non-essential but beneficial, and the remaining 8 are vital for not just human metabolism and growth, but are unable to be synthesized by our bodies, right? And so your question becomes, well, what's the difference between a non-essential and essential? An essential is we are unable to produce ourselves, whereas a non-essential, we are able to produce ourselves. Hence the non-essential and non-essential and essential, right? And animal proteins contain generally all 20 amino acids, all 12 non-essential amino acids, and all 8 essential amino acids. Plant proteins do not contain all 20, and a lot of times plant proteins are severely missing leucine, among all others. And leucine is a very important amino acid, especially for vision. It's vital for the proper, development of our musculature, and most plant proteins are missing out on that. So let's get into this. And what does the actual literature show? Well, I did a lot of research here for this because I wanted to bring up the most accurate information. And we're going to be talking about the digestibility and the ability of the human body to utilize these proteins. And so there were all of these studies looked specifically at humans to not only see if there was any remaining residue, i.e. amino acids, after being ingested by participants from various sources, and actually what percentage of these are being metabolized by the human body. And we're able to see that by radio-labeling proteins and specifically the nitrogens, and determining what the integration is of these proteins within human patients. And so what it has been determined is that beef on average has 92% digestibility and bioavailability. Black beans are at 0.75%, casein 1, so 100. Egg is 100%, milk is 100%, peanuts are 52%, soy protein extract is 100%, wheat gluten is 25%, and whey protein is 100%. And then if we compare this to another paper that was published 2022, last year, we can see that the trends that they reported are very similar. Milk, 100%, whey protein, 100%, beef is around 95%, soy protein, 100%, soy flour, 95%, pea protein concentrate, about 88%. Chickpea is around 74%. Cooked rice is even lower at around 60%. Cooked pea, 59%. Peanuts, 50%. Wheat, 49%. And whole grains at around 45% bioavailability. Now, why is this important? Well, this is important because what this demonstrates, or what this suggests, is that animal proteins are, for the most part, the only ones that are going to deliver a complete protein profile and 100% bioavailability. Now, whey protein, it did have 100% bioavailability. However, it's not a complete protein. And the other side of this is actual stimulation of protein synthesis. And so it was determined in several studies between 1998 and 2005, several of which were through the World Health Organization, that when looking at casein protein, despite the fact that casein protein had a bioavailability of 100%, it only stimulated protein synthesis at around 38%, and this was compared to whey protein concentrate, which also had a bioavailability of 100%, but that stimulated protein synthesis at 68%. So what was determined there is that where your protein is coming from plays a big role in not only the bioavailability of it, but the ability of the human body to use it for further protein production. And as with the casein protein of stimulating around 38% of protein synthesis, that consistently was rather similar with other protein sources, other plant protein sources such as wheat and peas and black beans. However, whey protein and soy protein were both relatively high, were rather comparable. Now, some of you may be wondering, well, why are we seeing a difference between the actual protein stimulation factor or protein stimulation effect of these protein sources and the bioavailability of them? The reason is because of oxidation. So some of these proteins, actually a lot of the proteins that are ingested in our bodies, go to oxidation for energy, to produce carbohydrates and to produce ketones, right? Now, ketone bodies are the primary energy source of the heart. However, we primarily get ketone bodies from fat metabolism. Nevertheless, they can still be produced from proteins. And the body likes to use proteins as an energy source. However, it sort of prefers carbohydrates just because it's easier to go from glucose to glycogen and to ATP than it is to go from protein to glucose to glycogen to ATP. However, a lot of the proteins that are ingested, in studies have been observed to be actively oxidized. So there were several other studies, this one titled Animal Protein versus Plant Protein in Supporting Lean Mass and Muscle Strength. And they determined that overall, collectively, animal protein tends to be more beneficial for lean mass than plant protein, especially in younger adults, due to the increased bioavailability of the protein and the fact that they are complete proteins compared to incomplete proteins from plant sources. So does this mean then that you can't get a complete protein source from plants? No. But unlike with meat, you can't eat one thing and get a completely balanced protein profile. You can eat a piece of salmon and it will contain all the 20 amino acids you need. But you cannot eat a piece of tofu and get all the 20 amino acids that you need. You would need to eat tofu, you would need to eat seaweed, you need to eat mushrooms, you need to eat protein from a variety of different sources with a strict plant source to make sure that you are achieving as close to 100% bioavailability as possible and to make sure that you are getting a complete amino acid profile. And in addition to this, there was another paper, The Role of Anabolic Properties of Plant versus Animal-Based Protein Sources in Supporting Muscle Mass and Maintenance. And they investigated if plant-based proteins have less of an anabolic muscle-building effect than animal proteins do to their lower digestibility and lower essential amino acid content, especially leucine, and deficiency in other essential amino acids such as sulfur amino acids or lysine. And what they determined was that ultimately, despite the fact that plant protein intake is doable, it is determined that animal proteins provide a much higher digestibility than plant proteins. And then they continue to cite the same data for protein bioavailability as the other papers we've looked at. So ultimately, what is the message with this particular paper? Well, they said that high quality protein optimizes protein metabolism at both whole body and skeletal muscle level, especially in older people and athletes. Plant proteins sources that are rich in fiber and micronutrients may be valuable, but they have a lower anabolic potential than animal-based proteins. Strategies to improve these properties would be by increasing protein intake, improving the quality of the protein, and mixing several plant proteins and plant proteins with animal sources to maximize your amino acid profile, your bioavailability, and the benefit that you are deriving from these substances. So that concludes part 2. I hope that by the end of part 2 here, you walk away feeling, like you know more about bioavailability, and that you feel comfortable being able to say to anyone who says it's a myth, no, it's not a myth, and here's why. As a brief summary of what we've talked about so far, before we go into the final part here, we've talked about the basic biochemistry of protein building, protein formation, etc. We've talked about the biochemistry of protein utilization, how stereochemistry plays a role, and how that affects bioavailability. And we've also talked about bioavailability, what protein sources have what bioavailability, why animal protein sources have consistently shown to be better for muscle building and overall protein and amino acid profile completeness than a strict vegetable or plant-based protein diet alone. And we have also hopefully determined how to better tailor your protein intake according to your physiological needs. And it is important for me to note that stereochemistry that we talked about before is the principal reason in causing these differences in protein bioavailability, is the fact that proteins from different sources will contain the same amino acids, but potentially in different enantiomers of those amino acids, either an L or a D configuration. So finally, moving on to the third and final part of this, we're going to talk about the 30-gram protein limit. Now, I did not know that this was a thing. Apparently it is. And if I just go to Google and I Google, 30-gram protein limit and see what comes up, the first headline will be from a pretty big hospital in America that begins with an M. I'm pretty sure you guys know what hospital I'm talking about. But they say general recommendations are to consume 15 to 30 grams of protein at each meal. Studies show higher intakes, those more than 40 grams in one sitting, are no more beneficial than the recommended 15 to 30 grams at one time. Don't waste your money on excessive amounts of protein. Now, that's of course not the only, place that talks about this 30-gram limit. Pretty much every website here will mention this 30-gram protein limit. How much protein can you ingest in one sitting? So this, I don't want to call it a paper. This article here was published for a website called Examine. I don't know what that is, but it was published by a gentleman who has a master's in public health and an MBA as well. And so he says that you should not eat more than 30 grams of protein in one sitting, because any more than that, and it is not digestible. Now, why is it not digestible? He says that the small intestines hold on to protein in what he calls a free amino acid pool, and that the body can draw amino acids from this as needed, on an as needed basis, and that if you ingest any more than 30 grams, because that is the limit of this, amino acid pool, it is not going to be digestible. So he continues to say that while it's not so simple, what happens is eating more protein increases your body's protein turnover, which increases urinary nitrogen levels, but it also increases a breakdown of your body's damaged or oxidized proteins. And so basically what he's saying here is, in short, eating more than 30 grams of protein results in wasted protein is incorrect, because it's not really wasted, it's just oxidized immediately. Now, is that accurate? Is it immediate? Is it where after 30 grams of protein it's immediately oxidized? Well, according to several studies, that does not appear to be the case, because it depends on the source of protein, or it depends on the source that this protein is coming from. In fact, several literature sources demonstrated that what actually happens is the idea that consumption of protein doses greater than 20 grams results in a greater amino acid oxidation is not accurate for all additional ingested amino acids, as some are utilized for tissue building purposes, and it is different based on the sources that these proteins are coming from. This idea that you have to stay under 30 grams of protein in one sitting comes from only examining fast digesting protein sources such as whey protein. Now, the question is, if you eat a steak, if you sit down, right? If you sit down and you eat a beautiful 6-ounce filet, or I guess an 8-ounce filet mignon, and on the side, you have rice and you have some peas there, maybe some legumes, and then you have a glass of milk. That's definitely more than 30 grams of protein. Is what's going to happen after you eat that. Once you go over 20 grams, is the rest of that just going to be immediately oxidized for energy? Well, the data doesn't suggest that. In fact, what the data suggests is that until you get to around 0.55 grams per kilogram of protein per meal, the vast majority of that protein is actually going to be utilized by the body for tissue building, assuming you are, exercising on a regular basis. Because it is important to note that this study that was done was in amateur athletes. It was in people who exercise on a regular basis. So I would say that the idea of sticking to 20 to 25 grams of protein a day would apply actually very well for the average person who doesn't exercise. But for somebody who does exercise, it seems like you'd want to be shooting for more of 42.5 grams of protein at the max per meal in one sitting, which would equate to around 2.2 grams of protein per kilogram per day spread out over 4 meals appears to be the best way to ingest your daily protein. And this is because up until you get to that 0.55 grams per kilogram per meal, most of that protein is going to go to synthesizing proteins to be used throughout the body. Once you surpass that, the rest isn't going to show a significant, benefit. So it's not that the 30-gram protein limit is a myth. It's more of that it kind of depends on the person. It's more of for an elite athlete, for somebody who spends a lot of time in the gym, that 30-gram protein limit may be more of a 42-plus gram protein limit. And for the average person, that 30-gram protein limit may be more of a 25-gram protein limit. And what the literature has demonstrated or is suggesting is that how much protein your body can metabolize in one sitting is actually more influenced on your activity level itself and what you're using your body for than just inherent dynamics of protein metabolism. And that was part 3. I know part 3 was definitely shorter than part 2 and part 1. I hope you enjoyed today's episode of The Science Behind That, and it looks like I'm at around 46 minutes here, so I was kind of accurate. I think I said 50 minutes. So ladies and gentlemen, I hope you enjoyed today's episode of The Science Behind That. If you have any questions or comments about this, please shoot me an email at thesciencebt@gmail.com. I would love to hear what you guys have to say, or respond to the poll right below the description of this in Spotify if you guys are listening to this podcast on Spotify. That being said, I will see you guys all on Friday. Have a great week, everybody. Thank you for listening, and remember to stand up and question everything.